in 1981, a group of scientists have performed a very interesting experiment on the coat color of rabbits. The results were as expected. 75% of the rabbits were white for the coat color and remaining 25% were yellow for the coat color. Now, it is matching with Mendelian ratio of 3:1. Now, another experiment was performed in 2022 with a separate group or by a separate group of scientists. The results were totally different. This time, scientists got 100% of the rabbit with white coat color. Now, the same experiment is performed 2 times with identical system. DNA sequence was same, there was no mutation, but in 1981 they were getting 3:1 ratio of Mendelian genetics, but in 2022 they were not getting the same. All 100% rabbits were white for the coat color. Now, what was the basic difference between these 2 experiments? The answer is interesting, and the answer also gave us the right definition about phenotype. Now, the major difference between these 2 experiments was in 1981 the rabbits were eating carrots, and in 2022 rabbits were eating biscuits. Now, if you look at a particular gene which is present in the rabbit, this gene codes for a particular enzyme which converts beta carotene into a colorless substance, or you can say into a colorless product. So, the 75% rabbits in 1981 were having this gene functional. So, when they were eating carrot, the beta carotene from the carrot was converted into a colorless product by the product of that particular gene or by the enzyme which was coded by that particular gene. But in remaining 25% of the rabbits, the gene was non-functional, it was not active, it was not performing, or it was not giving a functional enzyme. Because of that, in these 25% of the rabbits, the beta carotene was not converted into colorless compound, and that beta carotene was getting deposited in the skin of rabbit, and rabbits were yellow in coat color. Now, if we apply the same concept in 2022's experiment, we will find that since the rabbits are not eating any beta carotene containing system, or they are not eating carrot, they are eating biscuit. So, whether the gene or the enzyme is present or not, that doesn't matter. All the rabbits will be having white coat color, because even if the enzyme is active or not, the substrate of that particular enzyme, the beta carotene, is absent. So, in 75% rabbits which are having active gene product, beta carotene is not there, colorless compound is not there. In remaining 25% rabbits also, since they are eating biscuit, beta carotene is not there. So, there will be no deposition of beta carotene in the skin of rabbit, and the rabbits will be still white. So, we are getting 100% white coat color phenotype in 2022. Now, what has changed? The environment. Gene is there. The environmental change is what? Food. In one experiment, food was carrot. In second experiment, food was biscuit. We always learn about the definition of phenotype, and the statement says genotype plus environmental influence decides phenotype. This is the perfect example. In both the experiments, rabbits are having the same gene in same ratio, but the environment is changed. Carrots to biscuits. So, we are getting a different type of phenotype, a different phenotypic ratio in 1981 and a different phenotypic ratio in 2022. This is the best and perfect example to understand how the environment influences genotype to give different kind of phenotypes. Now, what do you mean by environment? Environment can be anything: temperature, light, food. And nowadays, when we are talking about cells, the environment can be the extracellular environment of a single cell. So, there is huge influence of environment or microenvironment on the phenotype of particular character, and that is why phenotype is nothing but genotype plus influence of environment.